What's that rotation?
Welcome, folks.
Zero. No.
Successfully made it once again to Science Colloquium. Before I introduce these speakers, are there any announcements?
So from now on, I think today moving forward, or maybe next week. Next week moving forward, Science Colloquium will be live streamed. So if we can't make it to the room, you can always check it out online.
No, you can make it to the room.
Yeah, that's true. Yeah.
You can be here.
Yeah, no extra credit, right?
Get your friends at other places to join you in Science Colloquium over live stream. That's very exciting. Any other announcements? Uneven still going strong?
Yep.
Yep, we're gonna
Yep.
make it.
Yay,
We're doing it. All right, believe in you.
Yay.
Believe in you.
Is it already on right now?
All right. Well, if there's nothing else, it is my pleasure to introduce Jeremy Smith to you. He comes today from Cornell University, where he is in his 5th year of the PhD process as a PhD candidate. And he's going to tell you today about his path into a PhD and about a couple of projects you're working on to give you a sense of work that's going on. Jeremy is able to stay after Colloquium, so if our Q&A section needs to extend, it certainly can, and we can stay in this room today to keep going questions. So please, have questions about careers in the process as well as the science, and we'll entertain them. If science immediately pops in your head, though, during the presentation, I think we can entertain a question or two during the talk, and there'll certainly be time at the end as well. Good? Awesome. Well, then please join me in welcoming Jeremy.
Hey everyone, I'm glad to be here. Thank you, Leah, for setting me. I'm in my 5th year of my PhD, and like Leah just said, I am talking to you about my PhD experience, but also diving more into some of the specific projects I've been working on, just to give you an exposure to what grad school might be like if you choose to go down that route. I'm going to start with a history of myself, and then talk about my time as an undergrad, my time as a graduate student, talk about 3 specific projects and collaborations, and then a little story about validating previously published work and why that's important, and then some tips for success and reminders. So first of all, I'm going to introduce myself. I'm from Buffalo. So now I'm at Cornell, so not too far of a drive for me. It's nice to have family nearby, but it's not always that easy for people. So I'm glad to be local. I'm familiar with the area, and I did my undergrad at SUNY Fredonia, which is on Lake Erie. It's on the border of Pennsylvania and New York State, if you know it. My bachelor's was in biology. I'll talk a little bit about my experience there. Finished my master's in food science at Cornell in 2021. I also currently doing my doctoral degree, my PhD in food science. Should graduate in May, and then hopefully after that I will be doing some postdocs. And my desire is to stay in academia. I want to be a teacher. I want to run my own lab hopefully one day. And some of my work that I've done as an undergrad and as a PhD student have led me to this path. I think it's important to mention that work-life balance is really important, especially in something like grad school. Most careers in general, but in grad school as well. Some hobbies that I have are, I'm a big fan of sports and music. I am both participating in them as well as just spectating. This is a picture of me and my dad at a Bills game, big Bills fans. And this is an instrument called ngoni that I made. It's an African gourd harp, a place called Gourdlandia in Ithaca. They grow gourds, and they have these workshops where you can build instruments. And so I just wanted to add that in to remind you that, while you might be working plenty of hours, it's very important to take time for yourself, and that's the best way to succeed in something like grad school. As an undergrad at Fredonia, well, first of all, I'll introduce Fredonia if you don't know it. It's a smaller school. It's somewhere probably in between Wells and Cornell in terms of student enrollment size. It's It's mainly undergraduate students, but there are some graduate programs. It's mainly masters, and the majority of the population is actually studying business and music. It's a big music school, but I went for biology and chemistry. During this time, not only did I have opportunities to take interesting courses, but I seeked out opportunities to join different labs and get research exposure. It wasn't until probably my senior year that I decided I wanted to have a career in research. I actually started pre-med, and I switched over after having to do a capstone experience which required lab, and I just fell in love with working in a lab. My first experience was an internship at a place called Timberfish Technologies. It's a wastewater treatment and aquaculture facility. That was in Westfield, New York, which is nearby Fredonia. This man here is Jerry Northrop, and he is the brains behind it all. He has a Ph.D. in biophysics, and he wanted to find an environmentally sustainable way to take a waste stream from a brewery and produce a product. So you're cleaning up the environment while you're also producing sustainable food source, just with the environment in mind. And there I had some experience with microbial ecology and water chemistry. From there, I joined a lab where it was, this is Courtney Wigdell-Perry. She was my undergrad informal advisor. She took on that role even though it wasn't her responsibility. And I studied nutrient limitation in freshwater lakes. Then I joined a second lab, which is not common, especially as an undergrad, but I had to take as many opportunities as I can to prepare myself for grad school. I knew I wanted to get into molecular biology and genetics, and I didn't really have opportunities to do that in these previous roles, so I joined another lab where we genetically engineered an algae to produce biofuels. I added this picture of me. Actually, if you're ever on the Fredonia webpage, on the biology program, I'm on the banner, and it's embarrassing, but it's also kind of amusing. Like I said, I want to teach one day, and so I TA'd organic chemistry. Having not, I'm a biology major, my PhD is centered around biology, but to be able to broaden your skill set and do something like chemistry is very important, and it looks good on a resume as well. That brings me to my grad school experience. I am in the Gibney lab at Cornell in food science, and Pat Gibney is my advisor. He comes from a background of yeast biology and genetics, not food science. So he kind of was put in this role to develop projects that are more applicable to food science, when in reality, its foundation is basic biology concepts. We kind of study the intersection of yeast biology and fermentation microbiology. And this was appealing to me because of those research interests that I have in the past, dealing with waste streams from a brewery, understanding fermentation microbiology, and yeast biology in general. I wanted to put up this list of projects. There's 8 here. And it's just to show you that a lot of people think you go into a PhD and you study one thing, you have your dissertation work to do, and it's, you become a specialist in one specific thing, but oftentimes it's not as simple as that. You have multiple projects going on. It's great because if one's not going well, maybe another's going well. And you also get exposed to new techniques in the process. It's not to say that it'll overwhelm you to have multiple projects. I actually think it's good to take on more opportunities. Today I'm just going to talk to you about the 3 that have stars, red stars there. But if you have any interest, if any of these things sound cool to you, I'm more than happy to talk about them as well. There's a prompt at the bottom, why did I pursue a PhD? I think it's important to also consider, because some of you might be considering going to grad school, but for different reasons. Ultimately, like I said, as a senior, I decided I'm going to go to grad school because I want to teach one day. I loved TAing, I loved doing research, but I didn't like having to always do what I was told. And once you get your PhD and you go start a lab somewhere, assuming the funding's there, you can do whatever you want as long as you can justify doing it. And so that was really the main driver. I wanted the independence to do the research that I was interested in, and not just what my advisor told me I could do. So now I'm going to start diving into specifics on projects. The first project is regarding diastatic brewing yeast. First, I'm going to introduce yeast in general. When I'm talking about yeast, it's Saccharomyces cerevisiae, brewing yeast, baking yeast, budding yeast, whatever you want to call it. It's kind of like the Swiss Army knife of microorganisms, because we know there are applications in the food industry to make bread, beer, wine through fermentation, but there's also other, and yeast extract, if you have heard of Marmite or Vegemite, spreading it on toast. You eat it that way, but also using yeast extract to make media for growing other microorganisms. But also, other than the applications that are more obvious in the food industry, yeast are a great model organism. We use them to study biological concepts, and this is because their genome is well characterized. We have plenty of sequencing data to defend that, as well as we know a lot about their genes that are in their genomes, and how, as a eukaryote, we can infer some of the findings in a model organism, such as yeast, to extrapolate to humans or just mammalian cells in general. And lastly, they're microbial factories, and this is relevant for all of these other points, honestly, because if they weren't able to produce ethanol, we would care about them for beer and wine making. If they didn't make tons of CO2, bread would not rise, right? And then also, yeast have been used and genetically engineered so that they can make some product, whether that's a company trying to make the medicine or an alternative food source to make, animal-free meats, such as impossible meats. So the term diastatic is probably new to a lot of you, and that's okay. I'm going to cover what that means. But my work is studying diastatic yeast strains, and how they differentiate from standard brewing strains is that they contain one of 3 genes from a family called STA. STA1, STA2, and STA3. And these genes encode for an extracellular glucoamylase. Don't get caught up on the words. Basically, it means they secrete out a protein that breaks down complex carbohydrates like starch, maltodextrin, things that normally yeast could not break down. And it's very unique to the subpopulation of yeast strains. And they're only found really in brewing strains, and that's because of the environment that they're in. They've evolved over time so that they can break down extra carbon sources for energy. So this secretion of the glucoamylase and subsequent breakdown of complex carbohydrates leads to more available fermentable sugars. And that ultimately leads to something called superattenuation in beer. Superattenuation is, you can think of it as depleting and fermenting way beyond the expectations of a brewer. And so this has some benefits and some consequences in the brewing industry. So like I said, it's a love-hate relationship with these yeast. Sometimes brewers intentionally use them. A lot of European styles use them to make dry beers that have fine bubbles and have different sensory components, such as beers like saisons. Most saisons are made with diastatic strains because they have that characteristic dryness to them. And that dryness is just that depletion of those complex polysaccharides that you normally would be able to feel in your mouth. But where I'm interested in diastatic yeast is the consequences as a brewer's enemy. And so if these yeast strains are contaminated into a beer, or maybe a brewery used them and had no idea this was a diastatic strain because it's a new strain for them, this can lead to a lot of spoilage results. And so it goes undetected typically because it's a slow process of breaking down these polysaccharides and then fermenting them. And usually it occurs in the bottle. And this is really concerning because as the bottle is a closed system, it pressurizes as CO2 is produced. And this can lead to foaming and gushing, in some extreme cases, exploding bottles. I've actually seen this firsthand. I've had a bottle, a can of beer in my fridge that had diastatic contamination, and the top blew off of it. And so it's prevalent especially in smaller breweries. And there's been lawsuits, breweries suing yeast distributors because this yeast must have been contaminated that you gave us, and we had to recall tons of beer. which is terrible for PR, but also, you know, on a small scale brewery, that's really problematic. So it brings some questions as to like how can we use research to resolve these problems. The main problem is that we found the presence of this gene stock does not predict spoilage potential by itself. It's an indicator that it's possible, but it's not a perfect yes/no. You can see that here. We have a panel of different strains. As I said, saisons are commonly used, common beers that use diastatic strains. These strains are associated with those beers. Doing a genotyping or just PCR searching for the presence of this genetic material, we can say, you know, these 3 strains have the gene, so they're probably a risk. But when you actually phenotype them and say how diastatic are they truly, there's a range between low or even non-diastatic to very strongly diastatic strains. And that's the main question that I'm asking. Why do we see this variability even though the gene is present? So the first step to resolving this issue is identifying strains that have different spoilage potential using an assay that our lab developed. So this assay is called the BGM assay. It stands for bromocressyl green maltodextrin. The components of the assay media are probably the most important, and it also explains the name. So maltodextrin is a carbon source that yeast cannot normally break down. And it's the only carbon source that's present, but diastatic yeast can break it down. So the only way a fermentation would occur is if the strain was diastatic. There's also a pH-based indicator of bromocressyl green, which turns from blue to green to yellow as the media acidifies. And as fermentation proceeds, the media acidifies, and the color changes from blue to green to yellow. So we look at this and we see the colors changing, we can say the strain's probably diastatic. And then we can use the rate in which this assay progresses to actually classify them based on their phenotype. Are they strongly diastatic? It's a real risk for spoilage. Or are they not really diastatic? So it's not really a big deal, and we don't have to worry about spoilage in the bottles. So this is an example of a strain that I took and subjected to the assay, and you can see it turns from blue to green. This is over a 21-day period, so it's a pretty long fermentation time, which pretty much simulates storage of the beer. Because the assay never completes and gets to yellow, this would be classified as low or non-diastatic, honestly. So we're saying, all right, it has the gene, but it doesn't quite progress the assay. Well, what else could be going on that's dictating this? And to understand that, we need to dive a little bit more in and separate some of the components that might be complicating things. And to do this, I wanted to pull out the actual protein that's responsible for this and see, is it functional? And to do that, I cloned the gene that encodes for the protein, put it on a plasmid, transformed the plasmid into a non-diastatic yeast strain, and subjected it to the assay. You can see here, in a situation where you remove all the strain background and you just are looking at the protein production, now it's strong diastatic activity. So what's going on in the background of this strain that's resulting in non-diastatic activity? The protein's functional, but we don't see it in real time. And this is probably to do with gene regulation, something that is, you know, transacting and not really a function of the protein itself. And to understand this, it's important for us to look at the DNA sequences to explain the variations. So the second part of this solution is to use DNA sequencing to explain the phenotypic differences between strains. Here are 3 different strains that I did sequencing on, and I'm showing it in a simple graphical way, but we can look at, you know, large data sets of DNA sequencing, whether it's the DNA or the, you know, translated protein sequence itself. And we were able to identify mutations. We have a standard wild type diastatic strain called French Saison, and we use it as the wild type for this example because it's the strongest diastatic activity. So we know it's probably the protein itself is highly functional, and the promoter, which controls the regulation of the gene, is also highly functional. So here on top, one has a wild type promoter, which I have in green, and a wild type open reading frame which encodes for that protein. So if you look here, you don't see any mutations. But when we look at this next one, it's a wild type promoter, but the actual gene has a series of mutations. And we can just see this looking purely at the DNA sequencing results. This last example here has a wild type protein sequence, but the promoter has a large deletion. And we wanted to know, do these variations impact the actual phenotype of the strain? And by looking at that BGM assay kinetics, we could see that a strain that has a wild type promoter and gene sequence has high diastatic activity, and you can see it actually transfers over into the protein when we pull that protein out, like I said before, put it on a plasmid, and check it without any of those complicating factors. When we look here at the second one, both the strain and the protein separated from the strain background have a reduced capacity to progress this assay. And this is because of the protein is consistent across both the strain and protein here. The protein is what is limiting this reaction and the progression of the assay itself, so we see a slowing in the assay kinetics. And this last example is a very cool example where the protein, which is wild type, has this strong diastatic activity, but the strain has no diastatic activity. Yeah?
I'm sorry. I just, because the beer is being bottled and sitting for so long, how does it, like, I'm trying to word this correctly, but even if it doesn't turn to yellow as quickly, it would still eventually become diastatic, right? Like, how does that affect, how does a lower level strain matter when it's going to become yellow anyway?
Yeah, no, that's a good question. So if there's any diastatic activity at all, even if it's weak, there is a spoilage risk.
Okay.
It just may take longer.
Okay.
And, you know, maybe a brewery can be familiar with those kinetics and say, in a month, there's a chance this could overpressurize. So maybe they have a best by date on their bottle to say, you know, consume this by a month, or put it in your fridge, which will lower the activity of the just general metabolic activity of strains.
Okay.
I would say it's extending shelf life. That's what they're trying to do, is extend the shelf life of it. That way, it doesn't go bad as quickly. I think that's the easiest way to do it.
Yeah.
Thank you.
Yeah.
So the third one, strain 3. So that one shows none of the activity, well the protein does. Just because the strain doesn't show any activity, does that make it fine, or is it because the protein shows the activity that makes it not fine, even if the strain looks fine?
Yeah, so I think that this strain is not a spoilage, right? Because the strain itself does not have diastatic activity over a 21-day period. Now, if I extended this 3 months, who knows what would happen, but, you know, oftentimes beers are expected to be consumed rather quickly, and so I think that this is an important point, because if we were to take this strain and screen for it beforehand, simply using PCR, looking for that presence of the gene, you might say, oh, this is a spoilage risk big time. But if you actually phenotype the strain, it's not. And so I think that's why it's important to do more than just simply looking at the presence of the gene, which is kind of the purpose of this, is to identify new diagnostic tools for breweries and yeast distributors to say, is it spoilage risk, and if so, at what extent? And how can a brewery deal with it? Maybe they could just ferment with this strain and then filter out the yeast. Then there's no spoilage risk. But a lot of the small breweries don't do that. So, okay. So yeah, that basically summarizes this project. At this point right now, I'm doing a lot more with looking at the phylogenetic relationship of strains, understanding how this gene even formed. It's such a unique gene, and it's hypothesized to have formed as a gene fusion, and only in a small subset of strains. So I'm looking into more of bioinformatically what's controlling not only the gene expression, but the evolutionary history of these strains. And ultimately, by providing a series of deletions that may be useful for breweries to say, well, now this is not a spoilage risk, we can develop better tools, diagnostic tools for identifying strains of interest. So I'm going to move on from that project if anybody else does. Yes?
I'm wondering if you've looked at anything like storage temperatures, right? Where it's like 4 degrees Celsius versus like room temperature. If you've looked at light, the influence of light on this, like if it's an amber bottle versus glass bottle. I know you're not working directly with the beer, you're working with the yeast, but I'm wondering if that will speed up or slow down these reactions.
Yeah, I would definitely imagine that temperature has a big role. But like I said, if you were to put a can at room temperature that has residual diastatic yeast and put one in the fridge, chances are the one at room temperature is going to explode first. The question is, does the one in the fridge explode at all? And my little anecdote about the can is actually partially not true. It was at room temperature when it happened. But so if you refrigerate your beer, chances are it's going to be okay. And if you buy beer from a big brewery, big beer distributor, you're not going to have many problems because they filter their yeast out. If you buy something from Aurora, there's yeast at the bottom of the can, and so they don't use diastatic strains. But if they had a contamination, you might see this. And the color of the light, the transparency of the bottle, I'm not sure if it plays a role. I honestly don't have a good answer for that, but it would be interesting to see if clear bottles, amber, green versus can where there's no light penetration, if you see different rates. I'm not sure. Okay, cool. And if you have any more questions about that, we can talk about it after. So now I'm going to talk about a second project regarding plasmid construction. Very brief introduction on plasmids. We have a bacterial cell here, and in these squiggles, this is the genomic DNA. This is a plasmid. The plasmid is this mobile piece of DNA that can go in and out of cells, and we can utilize them as tools for genetic manipulation in the laboratory. Plasmids contain elements that are essential for maintenance, for replication in a host, as well as for selection in a host. So this example here has an antibiotic resistant gene. And that will, if you supply the media with antibiotics, they can only grow, the host can only grow if it has this plasmid. We can also incorporate inserted genes here. Maybe you want to understand stop, for example. That's what we did. I put the gene in this region, and I was able to compare a panel of different STA genes based on using plasmids. So the purpose of this project is to develop a new plasmid set. And the reason we did this is because there's a sort of a problem in yeast biology in general where commonly used plasmids often require unique strain backgrounds for maintenance. And this is because of this right here, where if you have a strain that has a deletion in a uracil biosynthesis related gene, they're not able to grow on media that does not have uracil because they cannot produce it themselves, and it's essential for them to have uracil. So there's no growth on this media. However, you can utilize this mutation, supplement it by transforming in a plasmid with this complementary gene, and now this strain can grow on the media because it can produce uracil. But the limiting factor here is that the strain must have this deletion. Otherwise, there's no maintenance. There's no selection for this plasmid. So if we were to have a strain that doesn't have this deletion and it grew on this media, you don't know if it's because it's transformed or it just was able to grow. So to solve this problem where it requires a specific type of yeast to transform in these plasmids, we designed plasmids that have these more universal selective properties. And this is by using drugs, antifungal compounds. And if you were to grow pretty much any yeast on a media that has one of these common antifungal compounds, they're not able to grow. When you supplement it with the plasmid that encodes for the resistance to the antibiotic or antifungal compound, now they can grow. And this is important because it really does not matter. It's independent of the strain background. We don't need to have specific mutations. We just need to test it first. Is it sensitive in the first place to these drugs? Now we can add drugs to the media and transform in this plasmid, and we can expect anything that's growing has the plasmid. So what I did was I developed a large panel of 18 plasmids that had different elements on them so that it can be diversely used by any yeast biology lab that's interested. And they're actually on AdGene. If you are interested, you can access them and you can request them. They have selection and replication yeast elements. They have the origin of replication, which is required for the yeast to replicate the DNA. Two different versions, high copy and low copy. High copy meaning there are elevated levels of plasmid compared to the genomic DNA. And low copy meaning it's maintained just the same as the chromosome DNA. Also, we use 3 different drug markers for 3 different antifungal compounds. And this is important because, well, maybe your lab just doesn't have one of these to use. But also, maybe you isolated a yeast that interestingly has resistance to one of these. Well, we have 2 other options for you to try out to see, is your yeast sensitive? If so, then you can use these plasmids. And then at the bottom here, these are just pretty standard selection and replication markers for bacteria, which we use to to store the plasmids and produce a lot of it. And the result here is that I was able to transform this plasmid, the whole series, into a large panel of diverse yeasts. And I think that was the biggest takeaway from this project, was that it was diverse yeast. It was not just your standard brewing or standard laboratory strain, but there were brewing strains, there were wine strains of Saccharomyces. There were non-Saccharomyces strains, something like Lachantia guiveri, something really out there that not a lot of people have the ability to study because they don't have these genetic tools. So this provides us with an ability to now study these species more in depth because we can select for them and transform plasmids into them. Also in this process, I was able to gather data regarding their transformation efficiency. There's a variety of transformation, which is transformation methods, which is just where you take a plasmid and you stick it in the yeast cell. But the one we use is called lithium acetate. It's a very common chemical method to transform in DNA. And now we can say, based on our data, they have these different transformation efficiencies, which could, you can imagine, lead to more questions. Why? Why does Meshnicovia pulcherrima have really poor transformation efficiency compared to Saccharomyces cerevisiae? And there's an opportunity for people to now dive into these questions and say, what is there a genetic factor? Does it have to do with cell walls? Does it have to do with, I don't know, how the cell is responding to some of these chemicals? It's just a nice thing to see that one maybe simple seeming experiment could lead into a lot more follow-up projects. And the last project is going to be about the mechanism of methylene blue. Have any of you heard of methylene blue? By a show of hands. Yeah, so it's a pretty common viability assay stain, and we use it for bacteria and yeast, often in the food industry as well as in the laboratory, to identify and differentiate living and dead cells or viable and non-viable cells. Dead cells turn blue. That's the main concept of methylene blue staining, and that's pretty much the extent of the information we know about methylene blue. So the question is, why are they, maybe not so much why are they turning blue, but why are the viable cells colorless still? And it's hand wavy. It could be it could be either of those. Maybe dead cells are turning blue and viable cells are turning blue, but then they're getting rid of that color, and we don't really know why. So there are 3 hypotheses in the literature. That there's a reduced to colorless hypothesis where the stain gets into the cell, all cells, and only living cells are able to reduce it to a colorless compound called leukomethylene blue. And non-viable cells do not have this ability, so they remain blue. The second is there's an efflux pump, which is just a proton pump in the membrane that pushes out any compounds that the cell deems potentially toxic. And then the last is the intact cell membrane hypothesis, where only viable cells have an intact cell membrane, so it's impenetrable and the stain can't get in the first place. But compromised cell membranes, such as dead cells, are flooded with the stain. The problem is that there are all three of these hypotheses, but nobody's actually studied it. It's all just been predicted, hypothesized. And in the literature, people will claim it's one of the three, but there's no evidence. There's often not even a citation. If there is a citation, you can follow it back until it stops, and then there's no more citation. And that's a tricky thing when it comes to trusting previous literature. Is you have to, and I'll dive into that more later, but basically you sometimes have to do your research to actually understand, is this claim they're making valid? So how do I go through the process of saying which hypothesis is correct, if any? I could test them all individually, and I've done a little bit of this. I've seen that methylene blue is indeed reduced to a colorless form, but I don't know if that's what's happening in yeast cells. And I've deleted some efflux pumps, and it hasn't made much of a difference. So maybe a little bit easier and more of a broad picture view on what's going on, I could do something called a genetic screen. And the results are still TBD on this one. I'm still working on it. I've got a year to figure it out. A genetic screen is a very cool tool that geneticists can use to take this panel, a large panel, where there is a pool of strains, and each one has one unique deletion. And the deletion is, there's a barcode associated with it, so we could say what's there based on that barcode. And we take this population now and we subject it to methylene blue. Viable cells, we're expecting none of them to be blue. They should all stay colorless. The expectation is that maybe there's a deletion, a gene that's responsible for this conversion to colorless, that is not present anymore, and so now the cells, while being viable, are blue. So that's the premise. We can sort them out, look for the proportion of the portion of cells which are blue, and say which genes are encoded there, which genes are deleted that have a higher frequency of turning blue. And this is called barcode sequencing. And we do this by PCR and Illumina sequencing. And it's a very important question because it's something that a lot of labs use and we don't quite know why. Also, it's like a black hole in the literature. There's not any evidence. I've gone back to like the 1800s reading like these German papers, and there's no evidence. It's wild. But sometimes, I don't think it's laziness. I think it's just, it's a complicated question to answer, and you have to really invest a lot of time and effort into it, and hopefully in the process we're able to shed light on what the true mechanism is. Beyond my individual projects, grad school can be full of interesting collaborations, and this is just a list of some of them. I've worked with industry partners. Omega Yeast is a yeast distributor. I've worked with different people in my lab. I've worked with different people in different labs where they study very different things. This is a mycology lab in plant science department, and Cyrus V here is a food engineer. So I helped out with some very different things that have taught me some skills that I wouldn't have if I just focused on my research alone. And I was able to network in the process, get to meet people, and maybe get publications out of it, which is like the currency in academia. And so I'm going to just talk about a story about validating previous research. So, like I said, with methylene blue, there is a lot we don't know, even though people may say that this is how it is, but we don't have evidence to show it yet. So there are pros and cons when it comes to diving into literature. Literature is essential for you to understand how to develop new projects and how to confirm what's already been done and compare it to what you've done. So the pros are that it promotes new ideas. When you look through the literature, you read some papers and get some ideas. It's support for a concept that you might have, and critical analysis, you gain that expertise by reading a lot and saying, this is a good paper, this isn't a good paper, what did they do wrong, what would I have done differently? And you grow as a scientist when you do that. Some of the cons is it requires trust. And you can just blindly trust everything, or you can go and critically analyze it and say, I don't quite buy that. I'm going to do it myself. And just because it's published doesn't always mean it's factual. Often it is, but not always, because there's this trickle down of citations, like with methylene blue, that one person made this claim that it has to do with reduced to colorless with no evidence to support it, and everybody afterwards just said, yes, that's how it is, that's how it is. And then those citations get lost. So to not mistake published work for fact 100% of the time, it requires critical analysis. And so this is just one small story about a time where I encountered this in my Ph.D. So those genes that encode for that protein for diastatic yeast called STA, in 1988, the first publication regarding where they're located in the genome came out. And they said it was chromosome 4, 2, and 14. And they did this by running, doing something called a southern blot, which essentially they just run entire chromosomes down a gel and then search for their gene of interest and say, based on this ladder here, which chromosome is it probably on? And then I did a follow experiment. I wasn't actually trying to, confirm what they did. It was in parallel. It just was part of my work was looking at the DNA sequences. I took large chunks of DNA through DNA sequencing, and I aligned them to reference genomes to see where these genes fall, to determine is it STA1, STA2, or STA3, since they're all on different chromosomes. And I had this weird problem where one of them was not aligned to chromosome 14, which was the published location. And I found that all 10 of my strains that had this kept on aligning to chromosome 10, which made me question, am I doing something wrong, or was that previous published data from 40 years ago wrong? And so I dove into this paper a little bit more, and I found that, well, it's easy to make mistakes based on running jobs like this, because if you look here at the ladder, chromosome 14, which is where they said it was on, and chromosome 10 the bands are pretty much right on top of each other. And if you look here, it's kind of hand-wavy to say what it's on. But through DNA sequencing, we can confidently say, yes, it's on chromosome 10, but all the follow-up experiments pretty much just cited it as chromosome 14 without doing validation themselves. So that is the end of my research spiel. You can ask me any questions that you want, but I want to finish off with some tips for success and reminders just as a grad student to potential future grad students. I think it's important to choose a career that you're passionate about, especially with the Ph.D. I mean, you've heard this before, but with a Ph.D., it's 4 to 7 years of hard work. You're investing a lot of time. It's a little bit more than a 40-hour-per-week job, depending on your motivation. If you're not interested, you're going to be miserable. You need to join a lab or get hired at a job that you're very interested about, because it'll make life way more enjoyable. Finding a good mentor, this is specific for grad school, is key, because if you have a toxic environment and a toxic advisor, you might not enjoy the experience so much. So definitely reach out and meet these people before you decide, yes, I'm joining the lab or not. Also, don't just apply to labs because they're at, for example, Harvard or Cornell, and say, it's a Cornell lab, it's got to be good, I just want, that's not true. There are great labs at so many schools, and there are some not so great labs at some of the bigger schools. Talk to other students, you're not alone. Everybody that joins their first year feels the same way. And it really helps to talk to people and talk to people that are in their 4th or 5th year. because they can say, yeah, you'll feel that, but next year you'll feel great. Don't limit yourself to your narrow fields of study. This is what I said with my collaborations. I was able to learn about encapsulating cells and doing a simulated gastrointestinal tract delivery of probiotics. Never would have done that if I didn't do this collaboration. And I'm actually submitting a grant proposal soon that involves that technique. Would have never known it had I not helped out this lab. And maintain a work-life balance. This is critical. And if you feel yourself burning out, you got to take a few days off to ground yourself. Some last reminders is that you make progress every day. Even if it doesn't feel like it, you are. You'll look back and you say, I did a lot that year, even though it didn't feel like it at the time. You're here to learn. You're not here to get an A. I think that perspective is very important. Yes, you should go for an A, but if you are there with the goal of learning in mind, chances are you'll get an A and you'll retain that information along the way. Ph.D. is a marathon, but not a competition. You can't compare yourself to others. Everybody experiences it different. It's the same thing with undergraduate. You can't compare yourself to all of your peers because your situations are different. And then lastly, disconnect self-worth from academic progress. It's easy to get down on yourself if you are running gels and they're, doing a PCR and running a gel and it's failing every time. You think you're not a good scientist. That's not true. And I've found it's important to sometimes shelve things that you're struggling with and come back to it later with a fresh mind. So that is the end of my talk. This is a QR code to my lab page. You can see the projects we're doing. You can email any of us. I encourage you to reach out to me if you have questions, if you're interested in grad school at all. If you just want to know about my work or what it's like transitioning to grad school, I'd love to help. This is something that's important to me, is talking to students that are in the place that I was 5 years ago. So that's all. Thank you.
So I know some folks have classes soon, but if you'd like to stay and continue to ask questions, comments, happy to entertain some, but if you gotta go, you gotta go. But we will open the floor to additional questions. Anyone's got them? Grad school process, the science.
What led you to choosing Cornell? Was it just because you were also from New York, or what about Cornell that made you?
Yeah. Choosing the right school is important. For me, in my internship there were connections with Cornell, which allowed me to know what was going on. So it was kind of like a foot in the door, but in reality it just gave